I'm really thrilled to be able to present our keynote speaker. Our keynote speaker is Dr. Kohn from the University of California, UCLA. He's a distinguished professor of pediatrics, immunology, and molecular genetics. He's a pediatric bone marrow transplant physician, and he focuses on developing blood stem cell therapies for genetic disorders, including the development of the first successful bone stem cell gene therapy for a disorder called ADA SCID, as many of you might know as bubble baby. Babies are born without an immune system, and they generally die before the age of 2. And Don, I don't want to say single-handedly, because he probably won't let me say that, but almost single-handedly has cured, I use the word cure, over 50 babies with ADA SCID. And that is a single gene disorder that has been eradicated with this type of gene therapy. And so I'll tell you a little story about how we met Don and why Don's here, because he works on bone marrow transplant. And you might wonder why in the world we bring an immunologist transplant physician to the FAST Summit. And it's because in 2016, we worked to create a type of gene therapy called hematopoietic stem cell gene therapy with an incredible team at UC Davis. And we are high risk, high reward, and everyone told us it would never work for Angelman syndrome. But we saw the literature about brain diseases that were being treated with this type of therapy because it can cross the blood-brain barrier and get to all of the neurons of the brain. And there was no reason to believe it wouldn't help Angelman syndrome, outside of a lot of scientists not understanding the mechanism, therefore it couldn't be possible. And in classic form, FAST said, don't tell me something's not possible. We'll tell you if it's possible. And so we decided to fund this program thanks to the support of Dr. Dave Siegel, who brought to us a team that said, let's try it. And we did. And it completely cured the adult Angelman syndrome mouse model. For the first time, any therapy had actually treated every single phenotype. And we were so excited that we were going to be able to truly see a transformation like this. And we wanted to push it as quickly as we could to the clinic. But unfortunately, the year we took the license and decided to push it, news came out that a part of the gene therapy that we were using, which Dr. Kohn will talk about, actually was associated with some types of blood cancer. And we needed to change that part of the gene therapy because there were other similar ones that were out there that had this part, a promoter, that had a track record for decades of safety. And this promoter did not have that track record. And so Jennifer and I said, well, who can help us change this promoter? How do we figure this out? And we looked up the person in a press release talking about the promiscuous promoter, the expert in the world that had been doing this type of gene therapy longer than anyone. His name was Dr. Kohn. So in standard fashion, we emailed Dr. Kohn and said, would you be willing to work on Angelman syndrome? And his response was, sure, why not? And so here we are. It's been 2 years, and we finally have a human candidate to be able to push, hopefully, in the very near term, toward a human clinical trial. So with that, Don is going to talk to you about the journey of hematopoietic stem cell gene therapy.
Well, thank you, amazing Allison. Well, hello everyone. So yes, why am I here? Hopefully by the time I'm done talking, you'll get the connection between bone marrow transplant and Angelman syndrome, which is still hypothetical, but hopefully we could have a therapy that could be beneficial. I'm in multiple departments at UCLA. So this is what I'll talk about. I'll cover sort of a little background on blood and blood forming, hematopoietic, it's a word, will be on the test, stem cells and gene therapy. And then I'll talk about the work that Allison referred to, our work for many years on severe combined immune deficiency, that's bubble baby disease, and really to show you what can be done with this type of therapy. I've been at that for a long time, and now we have it working well. And then the main part of the talk will be about Angelman syndrome and how this can be applied possibly to it. And as Allison said, the data in the mouse that was developed at Davis, which you heard yesterday from Colin from my lab, we've completely been able to reproduce the exact same amazing findings in the mouse model, underlies the idea of bringing this to patients. So first, the part about blood and hematopoietic stem cells. So we don't all think about blood very much, but if we take a drop of blood and put it on a slide and stain it, these are the kind of cells that we see in blood. So there's the red blood cells that are basically packed with hemoglobin and carry oxygen. There's a different type of white cells that are mainly immune cells. And then there's the little platelets down in the corner that stop bleeding. So all of those cells come from the hematopoietic or blood-forming stem cell. And those are mainly for us in the bone marrow. And so on your left there, in the box, are the hematopoietic stem cells. They're sort of like the parent cells for the blood cells. And so over our life, that population of cells divides and self-renews and maintains a compartment, but then some of them go on to change and differentiate to make all the mature blood cells shown at the right. And so clinically, if we want bone marrow stem cells to transplant, the classical way is doing a bone marrow harvest. So it's an operation where the donor is asleep, with needles into the back of the pelvic bone, we extract bone marrow. That's largely been replaced now, although occasionally we do take bone marrow. Another source is the umbilical cord blood. So it turns out at the time a baby is born, they have these same hematopoietic stem cells in circulation. So if you capture the blood that's in the umbilical cord, which is the baby's, you can use that to transplant. Usually there's enough cells to transplant a child, not often enough for an adult. And what's most used commonly now are mobilizing the stem cells from the bone marrow into the peripheral blood and collecting them. I'll show you a diagram of that. So mobilized peripheral blood. And so the way I think of these cells is that the stem cells are the babies. They're a whole life ahead of themselves. They become any type of cell. Then in the middle are the teenagers that divide, make lots of noise, and then at the right are the adult blood cells that go to work, do their jobs. And just one other point at this point, there's a protein, a marker on these cells called CD34, and you'll see me refer to it several times. It's not exclusively on the stem cells, but it's on the stem cells and sort of the other early cells. So a lot of times when we're processing cells from a patient, we'll select those that have that marker just to enrich our targets. And so hematopoietic stem cells, or bone marrow transplant, has been around for more than 50 years. And mostly it's done for people with cancer or leukemia. So stem cells from those sources I talked about, bone marrow, mobilized peripheral blood, and cord blood, mainly it's for people with leukemia that didn't respond to the initial therapy. And so we need to give them higher doses, which will basically wipe out their marrow. So then they get the transplant as a rescue from that. So treating high-risk cancers and leukemia by allowing higher doses of chemotherapy than would otherwise be possible, hopefully to cure the leukemia, and using the stem cells basically as a rescue. So that's probably 90+% of transplants that are done. A small number are done when there's marrow failure. So there's a condition called aplastic anemia, where the bone marrow stops working. It can also happen after exposure to some drugs or radiation. But we're interested in number 3, and it can be used to treat genetic diseases. Basically, if there are blood cells or blood-related cells, by replacing the patient's genetically deficient cells with normal cells, either from a donor or, in our case, taking the patient's own cells and making them normal by adding the gene that they're missing. And this approach has been shown to cure a number of diseases. And so on the right there, might be a little smaller, are just a whole list of various diseases sort of next to the cell type they're involved with, where transplants have been done in patients and shown to be curative. And so by giving them a normal stem cell, they can make the red cells they need, for example, for sickle cell disease, or the immune cells for SCID. And on that list in yellow is Angelman with a question mark, so we don't know if it will work. But other diseases in that group, X-linked adrenoleukodystrophy, metachromatic leukodystrophy, the mucopolysaccharidosis, Hurler's, Hunter's, MPS III, all those have been treated successfully by giving bone marrow, and some of those bone marrow stem cells make monocytes that go into the brain that provide the missing enzyme. And that's the supposed mechanism by which Angelman would respond. And so the major barrier from transplanting cells from one person to another are the immune system. Unless you're going between identical twins, there are going to be differences that are detected by the immune system. So in a healthy patient, the normal patients, the recipient's T cells can reject the donor cells unless they get high-dose chemotherapy and immune suppression beforehand. But also the flip side can occur. Some of the donor's T cells that come along with the bone marrow or the mobilized peripheral blood in the new body they're in can see it as foreign and react against it. That's called graft versus host disease. So the grafted cells are attacking their new host, thinking they're just trying to get rid of something foreign. And as a clinical transplanter, that's what we spend a lot of our time dealing with, that complication. It won't happen from an identical twin. It's possible with a matched brother or sister who's a sort of a full match, they're still not identical, and higher if you go to a parent or an unrelated donor. And so then just sort of to step back and put into perspective what we're talking about, gene therapy overall is an area that over the last 30, 40 years has developed as a way of treating genetic diseases. And sort of the world can be divided into ex vivo, which is what we'll talk about, or in vivo. So in vivo is where you put the missing gene right into the body directly. And that's often done with AAV vectors that we've heard about, or you can put in editing reagents like CRISPR, or you can put in ASOs, to me sort of a type of gene therapy, where you're manipulating the genes by putting something directly into the body. And for example, the first approved gene therapy in the US was for an eye disease, where they put a virus carrying a gene missing in the cells in the back of the retina to restore vision. And this has been looked at, and there are drugs for muscular dystrophy going into the brain, into the liver cells. And the other half of the universe is ex vivo, where you take the cells out, manipulate them in the laboratory, and then return them. And so we can really only do that with the blood cells, the hematopoietic stem cells and T cells. And then sort of the hot new delivery method that's being looked at are lipid nanoparticles. So the same sort of material that's used to deliver the COVID vaccine can be targeted to be given like as an injection and go to a specific tissue and, for example, release CRISPR editing reagents. And so we're going to talk about hematopoietic stem cells as a target, but just sort of put it in perspective of where that fits in the whole field of gene therapy. And then just one other comment is that all these gene therapies we're talking about are to manipulate the patient's body cells, the somatic cells, but not the germline. We don't want what we're doing to get into the human gene pool. We're not smart enough yet to do that. And so part of getting reviewed and approved by the FDA is to demonstrate, if you're doing in vivo, that you will not get into the germline and have that be sort of a permanent change that could get passed along. And so sort of the hypothesis for this is that gene therapy using autologous, or the patient's own hematopoietic stem cells, provides a donor for every patient themselves. So you don't have to look for a donor. And it will completely eliminate those risks of graft versus host disease. And it also then makes the transplant easier. You don't need to immune suppress the patient before the transplant to prevent rejection or afterwards to control graft versus host disease. But as we've learned, you do need to make some space in the marrow for the cells to engraft, and that's called conditioning, and I'll talk about that because that's kind of one of the biggest issues involved. And so, from an engineering standpoint, the technical challenge is to get the normal gene into that long-term stem cells, because those last a lifetime and can be transplanted and will last a lifetime in the patient that gets it. All the other cells to your right of that are sort of short-term cells and will last weeks or months, but won't be permanent. So a lot of the early learning in the field is what are those stem cells and how do we manipulate them? And the way we do it are either adding a gene, like with a virus that I'll talk about today, or now we can also edit the genes in the stem cell with things like CRISPR, where we can fix a mutation, and then it will be passed along to all the blood cells that will be made. It's estimated that each stem cell, once it starts dividing, makes literally billions of other blood cells. And if you get the gene in it permanently, that will get passed along to all the cells that are made. And so just a little about the gene delivery tool that we're using, or vector. So for stem cells that are going to divide, you really need to get the gene to go into the DNA of the target cell, so it gets copied every time the cell makes a new copy and gets passed along. So AAV does not do that. And so AAV, although it's efficient delivery, it doesn't get into the DNA, and so it would get diluted out once the cells start dividing. And so we've mainly used a class of viruses called retroviruses, and one of their submembers is called a lentivirus. So in the laboratory, we clone the needed gene into the virus, package it into a viral capsid, and then add it to the cell. So that sort of arc is the membrane of the cell. Then the genetic information in the virus gets copied into DNA and goes into the chromosome and integrates permanently. So it becomes just part of the double helix. You've got this little gene unit that you've added, and for example, it could be UBE3A expressing that gene. So in fact, there's UBE3A going into the DNA. And so this technology was developed sort of in the middle 80s, about, it's a shame to say, when I came into the field. And it's really been developed to a point where it's really very quite efficient now at modifying stem cells. And so this is, again, sort of another cartoon of the process. So we start with the patient, we collect their stem cells, and then in the laboratory, we can add a gene from the top with an integrating virus that will go into the DNA of the cell and be there permanently, or coming up from the bottom, we can edit the cells now with CRISPR. CRISPR is generally better for sort of fixing single mutations, but adding a gene will override any mutation of that gene. And then before the cells go back in, the patients will get some conditioning chemotherapy to make space in their marrow. So we give the cells back, they can land and grow, and I'll show you a cartoon of that. And then the cells are given back to the patient. And the transplant is actually the easiest part. It's not a surgery, it's just an infusion of cells like a blood transfusion. And so this is showing a person giving mobilized peripheral blood. So they get a growth factor, GCSF nupogen, that makes the white cell count go up. Turns out it also causes the stem cells to leave the bone marrow and go into the bloodstream. So after 5 days of GCSF, sort of boosted on the last day with another medicine, plerixafor, there's many stem cells in the circulation. So all we need to do is collect blood from the patient. So this process called leukapheresis, some of the patient's blood goes out through an IV line, goes into that centrifuge that collects the stem cells, and everything else goes back to the patient. So over the course of about 3-4 hours, we've taken their entire blood volume through the machine 2 or 3 or 4 times and skimmed off all the stem cells, but returned everything else to them. So when they're done, they still should have all their other blood cells and not really have a problem from it. So this is done usually in a clinical setting. For kids, we bring them into the hospital and do it in the hospital. And then you get what's shown in the bottom right, a bag of basically white blood cells that are very enriched for stem cells. And so then this is showing the processing. So upper left is the patient getting leukapheresis in the unit. Then they go to what we call GMP, or Good Manufacturing Practice Lab. That's the ultra clean room, like where you could make computer chips, or we're making cells for patients. So we don't want any risk of contaminating their cells with, the bacteria that are on the workers. So everyone's gowned and gloved and masked, and the air is ultra filtered, et cetera. The stem cells are brought there. They're processed to isolate the CD34, enriching for the stem cells. And they're cultured for 2 days typically to add the virus with the missing gene. And one day that will be UBE3A. And then we freeze the cells. So they're only in culture for 2 days. We freeze them down, and besides the main bag of the cells, we have several vials of the same material that we can then thaw to test to make sure every cell took up the gene, that it's sterile. And so we have a whole list of criteria that are listed there. And the cells have to pass 100% of their criteria to be able to be used. And they're released by the quality assurance unit of the GMP, and then they're ready for the patient. And then they're just infused as shown there through an IV. And so the elephant in the room, I think, in transplant is conditioning, as we refer to it. And so conditioning is where we give chemotherapy to make space. And in different types of transplants, we'd either sort of give full condition, where we really try and wipe out basically all the bone marrow, so all the cells that grow afterwards come from what you gave back. Or in some cases, like SCID, we only need to do that partially. Or in some cases, we don't do that at all. So here's my Los Angeles sporting college team analogy. So if your marrow is filled with all these USC footballs, and then either you give no conditioning, full conditioning, or reduced intensity, that's kind of what you have afterwards. So if you give no conditioning, there's very little space in the stem cell niche. If you give full conditioning, you've really opened things up for cells to engraft. And if you give sort of an intermediate amount, you open up some space. And now we give the UCLA basketball stem cells. And so with no conditioning, there's very little engraftment of the cells. And we actually had a trial in the early 2000s where we started out not giving conditioning. We didn't get engraftment. This is again for SCID patients, then we started giving conditioning and everyone engrafted. So in that study, we kind of proved, at least in that setting, it's essential to make some space. And if you fully ablated the marrow with high-dose chemotherapy, then you basically, it's taken over completely by the cells you've given back. And that's what I think we'd want to do for Angelman. But this is, again, probably the most difficult part of a transplant, the conditioning. So the conditioning currently used prior to infusion of the gene-modified cells is a chemotherapy drug called busulfan. And it's given over 3 to 4 days before the transplant only. So it's not like multiple courses of chemotherapy. It's just 4 days of getting this drug twice a day typically. And that will completely wipe out the bone marrow stem cells. And actually, the patients are usually okay the days they're getting it. So it's kind of different than if any of you have experience with a relative getting chemotherapy. It's really over the next few weeks that they will have the effects of that. So some of those effects are listed there. So they may get a condition called mucositis, where the mucous membranes of the mouth are irritated because they're a dividing cell, so the chemotherapy hits them. And so often the patients for a week or two won't really be able to eat and will need to get intravenous feeding, might need pain medicines. So that's kind of, again, the hardest part of it. But people get through that, but it's challenging and certainly may be more challenging in patients with Angelman. Typically, from that amount of chemotherapy, the hair will fall out in a week or two, but it comes back. But again, for some patients, that's very upsetting, obviously. There may be nausea and vomiting, although we really have very good drugs now to control that. So usually there isn't a lot of that. Rarely the drug can cause liver damage. Mainly we see it in people who've had a lot of chemotherapy before, like for leukemia. And one of the other side effects that may cause infertility, because the egg-forming cells and the sperm-forming cells are also sensitive to the chemotherapy. And so there are ways in advance to preserve egg or sperm for future use if interested. And then there's hypothetically low risks of late cancer or lung problems. We haven't seen that really very commonly, but it's at least a risk. And so that's, again, I think it's important to really address that because that's the hardest part of a transplant. So that means getting your child through a couple of weeks in which they're not very happy, they need medicines, they need intravenous feeding, meaning they'll need to have an intravenous line that whole time. There are a lot of new things coming along, ways to replace the chemotherapy with antibodies to stem cells to eliminate them without really those kind of complications. They're not quite there yet. It's promising. Then there's also sort of the challenge of getting it commercially approved. So I think in 5 years or so, maybe we'll be able to stop using chemotherapy. So then just sort of more about the transplant. So on your left, that's Nurse Lindsay bringing Ava's transplant bucket. So every child before their transplant, we fill up a bucket with all the tubes and connectors and everything they might need. In the middle is that tank that looks sort of like R2-D2. That's a liquid nitrogen transport tank that has the stem cell in there. And then on the right is the thawed stem cell bag. And so you can kind of see there's an air fluid level. So the little bit of cloudiness below that, that's the stem cell graft. So that might be 100 million or 200 million stem cells. And that will be enough to repopulate their bone marrow and make their blood cells for the rest of their life. So that's kind of the most amazing part, I think. And so here's a patient who had SCID getting his transplant. And so on the left, you can see that syringe is kind of cloudy. Again, those are his stem cell graft. The tube on the right is just the saline flush. So those cells are just over 5 or 10 minutes. They're slowly pushed in through their IV line. They get into the body. They go through the body, and they'll make their way to the bone marrow and stick and start growing. So that's sort of the background on stem cell transplant. And so now I want to talk about our experience with severe combined immune deficiency to show you what can be done and what the results can be. So this is a famous child for those of you who are of my generation. This is David Vetter, the boy in the bubble. So this was a young man who was born in Houston, Texas in the early 70s. His younger brother had just died of the recently described disease called SCID. So at birth, he was put into isolation, tested, and found to have it. And he was tested, and his one sister was not a match. So there wasn't a way to transplant him. So he was kept in a sterile environment for 13 years. So it was very much in the media because what's mankind come to that were keeping people in bubbles. And eventually he had a transplant from his sister and died of complications at 13. And he had that disease, severe combined immune deficiency. And so we've worked on this disease, again, since I've been working on it since the mid-'80s. And this is the work, since I've been at UCLA, of over time, all the patients we've transplanted. So we've done multiple trials, iteratively, trying sort of version 1.0, seeing what worked, what didn't work, coming back with version 2.0. And we're sort of currently in version 3.0, which is on your right, which is using a lentiviral vector. So lentiviral vectors are what are mainly used now for putting genes permanently into hematopoietic stem cells. And it needs to be said, they're derived from the HIV virus. That's lenti is the class of viruses that belongs to. All the virus genes are removed and replaced by the gene to be carried. And they've never caused a problem like causing immune deficiency like AIDS, but they're actually very well adapted to getting into human cells. So we've done a number of trials with that virus that I'll tell you about. So that's the map of the virus. So let me kind of walk you through it. So in the middle is the human ADA gene. And we've tweaked it, we've coded and optimized it. So there are different letter combinations like AAA and AAU and AAC that code for the same amino acid to be put into the protein. And cells have favorites. And so if you change the sequence to have the favorites, you make the same protein, but you make it more efficiently. So it's coded and optimized ADA sequence. And then the promoter in front of it that, as we heard yesterday from that great talk by Dr. James, is sort of the control unit that drives it, is from a cellular gene called elongation factor, short. So it's a promoter that will drive production of the gene, but it can't interfere with nearby genes. And as I'll show you, we have a great deal of safety data. And so we actually developed this jointly with colleagues in London. And so over the last decade, we've done trials in parallel, same batches of vectors, same protocol. And I'll show you, as of now, we've reported 62 patients treated. This was a little earlier when we did trials both in London and at Los Angeles. So this is sort of the patient journey through the gene therapy. So the first part is talking to the family, discussing risks, alternatives, et cetera. And if they agree, having them sign the informed consent, then having screening tests just to make sure they meet the criteria. And it's things like echo, EKG, blood tests, just to make sure they're healthy enough to go through the transplant. If they are, they then come into the hospital for the stem cell collection. So they'll get injections 5 days in a row of GCSF, they'll get a dose of the drug plerixafor, and they'll get leukapheresis for 1 or 2 days to collect their stem cells. Then they go home the next day. The cells go to the laboratory for doing the kind of processing I talked about. So we isolate the CD34 cells, we add the lentiviral vector with the normal ADA gene and freeze them down, or cryopreserve them. And then it takes about a month or so to test them to make sure they meet all those criteria. I talked about sterility, took up the gene, et cetera. And then when we know that the cells are good and they're signed off by the QA unit, the patient comes back for the second admission. And this is the transplant admission. So they'll come into the hospital, they'll get a central line placed, either a long IV line, a PICC line in their elbow, or actually a surgically placed central line that can stay in for months and are quite secure, although I have seen 1 or 2 kids pull them out. And then starting that day, we'll give them their chemotherapy. So it's showing busulfan on Monday. And it turns out that the metabolism of busulfan differs a lot person to person. So we actually personalize it. We measure how quickly the person cleared that first dose, and we adjust their later doses to try and have total area under the curve or exposure to the drug. And so we've gotten much better at targeting it precisely. And then on Friday, so it started on Monday, on Friday they get their cells. So again, we bring the cells to the bedside, we thaw them, and we infuse them. And they're kind of done with the transplant, and hopefully it will have an effect. But then they have that few weeks afterward from the side effects of the chemotherapy. And this was the frozen trial. This is the first time we did frozen cells, so we call it the frozen trial. And so we've done this now for around 50, more than 50 or 60 patients with this lentiviral vector. We did some earlier ones. And this is just a map of where they came from. So rare diseases like this, the patients are not all in Los Angeles. So they're all over, and we basically had patients from the US and Canada, so I actually got better at relearning my Canadian geography, which I learned in 3rd or 4th grade. So there's arrows from the different trials showing where the patients came from. And so we would bring the families out to Los Angeles for the stem cell collection. They would go home, they would come back for the collection, and while they're there, we'd have the patients stay at Ronald McDonald House or a hotel like that. And usually in our transplant unit, one parent can sleep in the room overnight. And so we reported our results, this is now 4 years ago. This was a report of 50 patients, 30 treated by UCLA, 30 at Great Ormond Street Hospital in London, with this approach that I talked about, using CD34 cells, that lentiviral vector, lower dose busulfan. And what we reported was 100% of the patients were alive and well, and they still are to the present time, and 96% of them were event-free survival. So we just define an event sort of it failed, and they need to go back and have a transplant. And so we had 2 patients in the initial group where the cells just didn't grow, and they needed to go back on the enzyme, and then they had a transplant later. And then so we just had another paper come out 2 weeks ago, sort of following that cohort plus another 12 that were done in London sort of a year later. And so in this paper, we reported 62 patients with ADA SCID treated again at UCLA and Gosh between 2012 and 2018. So now we're reporting how do they look a decade out, basically. So the time we analyzed the data was 2023, so actually they're 2 years beyond that point now. And what we were able to report was that it was completely persistent and stable. So all the patients had stable, when we measure the gene in the blood cells in the circulation, we measure their T cell numbers, et cetera, no change after the first 6 months or so, and really quite good. They had good immunity. There were no complications from the gene therapy or from the vector in any of our patients. And then this is just one graph, it's kind of small, but just shows different T cell populations that we measured over time are all stable out to 10 to 12 years. And so we hope this will be stable for their lifetime. We don't know that yet till we follow them for their lifetime. But it really looks like this is persisting, and so that's obviously very important to have a long-term effect. So the stem cells that we took out of the body, put the gene into, put back, are still now in the bone marrow making blood cells. And in fact, at least 18 different blood cell diseases or genetic diseases have been treated by this approach. So this is a list that I sort of keep myself of all the diseases that have been reported in clinical trials or at meetings treated by this approach of lentiviral vectors, putting the missing gene into hematopoietic stem cells. And so on the list, the top 2 on the left are genes I mentioned before, adrenoleukodystrophy and metachromatic leukodystrophy, which are neurodegenerative disorders. And by giving them gene-corrected bone marrow and getting cells going into the brain, they make the missing enzyme and really stop the neurodegeneration. And on the top right, MPS 1 and 3 are also similar diseases like that have been treated. And so Angelman has not been treated by a bone marrow transplant or by gene therapy yet, but if it follows the rules that these other diseases did, it should be beneficial. So in all these trials, lentiviral vectors have had an excellent safety profile, one exception being actually an ALD, and Allison referred to this. That virus they used had a mouse retrovirus element driving the gene, and those can cause a problem. And coming soon, we're working on alpha thalassemia, but we're also working on Angelman syndrome, and hopefully they will be on this list in a few years as having been successfully treated. So let me talk now about how this would apply to Angelman syndrome. And so this is another view of the hematopoietic stem cells making blood cells. And the important thing is on the left, one of the blood cells that's made are monocytes. And monocytes are cells that mainly become macrophages or big eaters. And they're in tissues, they're in the blood. Some subset of those monocytes cross the blood-brain barrier and turn into microglial-like cells. The aficionados don't allow us to call them true microglia because those only happen in the embryo, but they functionally are microglial cells. And then this is sort of a little more complicated version. So on your lower right shows that in utero, cells from the bone marrow go into the brain and form the microglial cells that you're born with. But postnatally, after you're born, cells from the bone marrow can make monocytes that can go across the brain and make microglial-like cells. And that's accelerated by a bone marrow transplant. And in fact, the busulfan, besides increasing engraftment into the bone marrow, also increase engraftment of microglial cells into the brain. And then the other aspect of this is cross-correction. So the UBE3A gene that was developed by FAS with the group at Davis is designed to actually have the protein be secreted from the microglial cells so it can be taken up by the neurons. And I think that's kind of the secret sauce of this. And so if you get these microglial cells that came from the stem cells with the gene, they actually can release the protein and cure the neurons around them. And so actually Colin showed a version of this yesterday where the procedure then is collecting the stem cells from the patient, adding the virus with the UBE3A gene, transplanting them back in, and over time they will then make microglial cells in the brain. And so this is a slide from the work done at UC Davis, and they really pioneered this work, and we're kind of really riding in their wake. So they made a lentiviral vector, and this was the one made from mouse, so it has the mouse UBE3A gene, and it uses that retroviral promoter to drive expression of the gene. So it's very strong and make a lot of the product. And they did very nice work in the mouse model, again, as Allison said, curing all aspects, behavioral. That's the graph on the bottom right is delta power, showing that it's hard to read, but the delta power in the transplanted mice was as good as in the neurotypic wild-type mice. But the problem was the promoter element they used is the one that was shown sort of about as this work was being done in ALD to have risks of triggering leukemia. And so, and this is a paper describing the ALD studies where patients were cured of their disease by the gene therapy, but over time, an increasing numbers of them developed sort of an early pre-leukemia, and some of it went on to leukemia because that viral element turned on cellular growth genes. And that's kind of been known as a risk, and so most of the field has moved away from using that, and instead using like we use for the ADA that has a very good safety record. So our primary goal when Allison first came to us was to make a vector that would work as well as the other one, but that was safer. That would have control elements that wouldn't risk turning on genes near where they land. And then ultimately that comes to now translating this to a clinical trial of gene therapy. And so the work that I'm going to present now was done by Roger Hollis is the lead scientist, Christopher Luthers, who spoke here last year and who is amazing, started this work, and then Colin Jewett, who spoke yesterday, has been carrying it on since Chris finished his PhD and is at the CDC to become an epidemiologic intelligence service officer, which will be great for the world, but we miss him. And so they made a series of lentiviral vectors that have the UBE3A gene in them and use different promoters. So we made one like the Davis one with the M&D promoter as our sort of comparator, or then we used one with the EFS promoter that's in our ADA vector, or another promoter, PGK, that's also been used in a number of clinical trials and been shown to be safe. And so our question was, can we make one that will work as well as the M&D one with a better safety profile? And so this is a Western blot showing the level of expression of UBE3A protein, are those bands, and the two on your left are our new modified vectors. And again, we did that trick of codon optimizing the gene so it makes more protein. And by doing that, we're actually making more copies of more amount of the protein than the one with just the M&D promoter driving the wild-type gene. And so then we went out to do a number of studies, again, following what Davis had done and guided a lot by Allison and Jennifer on these studies, taking these different vectors, putting them into the mouse model, and then doing behavioral and EEG and histochemical studies. And I'll just show you a little of it, because again, Colin presented this yesterday. So we used either the EFS-driven promoter, the PGK, or we didn't modify the cells. We just mock transplanted cells that still were not corrected, or we took bone marrow from wild-type neurotypic mice or from the Angelman mice. So we had 5 groups of mice. This graph is just showing the level of engraftment. So the panel on the left, we actually always used male donors into females so we could measure how much cells are engrafted by measuring the Y chromosome for the male cells. And so the groups at the top were the ones that in the blood are the two different vectors, PGK and EGFP, and EFS and PGK. So the bands in the middle are in the blood and the bone marrow. So we have 80–90% engraftment of the donor cells. And then the lower left of that panel are measuring the male cells in the brain. And so they're about 100-fold lower, but that's because most of the brain has other cells in it. And then on the right is measuring the vector copy number. So we have about 1 to 2 copies of the vector in the blood cells. And again, the brain lower, but still about 0.5% or so of cells with the gene in the brain. And then we saw all the same kind of amazing behavioral improvements. So this is on the left, open field that measures sort of their mobility, and the lower 2 bands are the untreated Angelman mice or those that got just Angelman marrow, and the bands above it are the ones that got either the wild-type marrow or with either of our vectors. The rotarod assay that we saw a nice video of yesterday shows that the mice that got gene therapy performed as well as the wild-type mice in terms of their ability to stay on the rotating rod. And then these are other measurements of cognitive function, novel object recognition. And again, the gene therapy mice performed as well as the wild-type mice, as Davis had shown with their vector. And then here's the EEGs. And so sort of our conclusion from the mouse studies that, again, Chris presented last year when we were sort of midstream of those, and now Colin has finished up and we have a paper that's under review. Both the EFS and the PGK-driven vectors had improved expression over the potentially oncogenic MND vector. The treated mice showed improved motor and cognitive function during behavioral assays. The treated mouse delta-powered measured by EEG was reduced to wild-type levels. And the immunochemistry, which I didn't show, but Allison actually showed some very nice pictures yesterday, on the brain slices showed that the protein is being made in the brain, and we can actually see it in the neurons, so presumably it's leaving the microglial cells and being taken up by the nerve cells. And so the question is, mice are mice, and can the excellent benefits that we see in the mouse models, will that have any bearing on what we see in patients? And again, the mice that we treated, they were transplanted about 6 to 8 weeks of age, which is sort of like a late teenager in human development. And so they weren't infant mice, and so at least at that age, there was benefit that persists. We usually study them about 4 months later. And so with FAST and with Transformatics, we applied to the California Institute for Regenerative Medicine, our stem cell funding agency in California, for a grant to carry this forward. And so the grant is a TRAN1, which is a translational grant. And our goals are to start off by having the vector that we made in our laboratory made under commercial standards, so it would be suitable for human use. And that's going on right now. And then that same manufacturing company will be the one that will make the cells for the patients. Then we need to do various tests for safety, so put the cells into immune-deficient mice, for example, and make sure they don't cause any complications. We'll need a clinical protocol, which is drafted, but we need to sort of finalize that, the informed consent document for a trial. And then we would meet with the FDA. And so to do a clinical trial, any new substance, you need an investigational new drug, or IND, permit from FDA. And FDA is very helpful at letting you meet with them in advance. And so a pre-IND meeting is when you meet with them and tell them what your plans are, show them the data you have so far, and they will tell you, is that enough, or what else you need to do to apply for approval. And so we'll have a pre-IND meeting. We'll do whatever studies additional they want us to do, and they would submit that application to the FDA to do a clinical trial. So the hope is to open up a trial for this within a few years. And so I'll just leave with some questions. There's a lot of unknowns, obviously, and as a physician and as a scientist, I try and, deal with reality. And, so I think there's a lot of things we don't know. We know it works well in mice, but for example, first of all, will it be feasible for Angelman patients to undergo this procedure? It means about a month or more in the hospital with IV lines and going through and recovering from the chemotherapy. So it'll be a challenge for the patient and the families. But we've done other people with disabilities and gotten them through the transplants. Will it be safe? Are there unique problems inherent to Angelman that might make it more risky? And then ultimately, will it be effective at improving their clinical features? And that's what we're hoping to see. So I will stop there and just show you, this is my lab group as of last summer, so we have a little army of people working. Colin's way in the back of the picture. And then this is our acknowledgement slide. So again, the work at our lab, Roger Hollis is leading it. Chris Luthers did a lot of the initial work. Colin is finishing up with a number of other people assisting him. We've had a number of labs at UCLA that assisted us. And then we've done this very much as a partnership with FAST. And then we have the recent funding from CIRM that hopefully will let us take this to the clinic. And I'll stop there. Thank you. Oh.
And we'll have time for questions to all of you present.
Sorry about that. Thank you so much for such a great presentation and really, really important work. Obviously, all individuals with Angelman syndrome are missing a functional copy of UBE3A. But as we heard yesterday, many with the deletion genotype may also be missing or haploinsufficient for many genes. If this is proven to work, do you foresee a future where maybe individuals could receive an individualized set of genes for those that they're needing?
Yeah, so it's very hard to hear up here, this echo, but I think the question was about, would this work for people with deletion? If was that the main point of the question? Can they get the other genes? So, yeah, I don't know that, and so this is my first time here, so that's a question that obviously I've been thinking about a lot since being here. And a question is, should we start just with people with mutation and then see how that works? We also need to get the mouse with the deletion and test if you just replace the gene. If you need to replace the other ones, then you could probably put 2 genes in the same vector. But if you need multiple genes, it might be more challenging. There are techniques being developed to put larger pieces of DNA into cells, and they work in the laboratory, but not very efficient for primary stem cells. So I'm not sure how that would be approached. And hopefully the research will hone down what are the other critical ones in the deletion area, and maybe 1 or 2 of those could be added at the same time.
And something else just to add related to that is that we all know UB3 is the major gene that dramatically impacts phenotype. The other genes may contribute, but it also might be marginal. So I think with all the therapies being developed for deletion or any genotype, UB3 replacement is the first step. And then dealing with the other genes, maybe a different modality, an ASO, a CRISPR editor, a different technology that might be piled on top of something like this. I don't think this technology will solve all of it, certainly not for the next 10 years. One question back there, and then we're going to move on.
Okay.
Okay. I was just wondering, would this work for patients with UPD?
Do you want me to answer that?
Yeah.
Okay. Yes, so just like any gene replacement therapy, any genotype that is loss of function, meaning not producing, making, or expressing UBE3A, which would be deletion, loss of function mutation, UPD, or ICD, or even mosaic, would all benefit potentially from replacing UBE3A. So this would likely be genotype agnostic outside of mutations that could have any gain of function or dominant negative effects. So it would be any loss of function genotype, similar to the AAV gene therapy that we'll be talking about next.
Okay, thank you.
We'll ask more questions on the panel and certainly reach out throughout the breaks. Feel free to walk up to Dr. Kohn and tell him all about your child.